Thank you, Gabriella. Thank you very much for the invitation. And I'd like to say I'm honored to be here. This seems like a really excellent meeting, and I'm very excited to be with you this morning. So I've spent my whole career working on blast diseases. And you might think, well, maybe that's boring, but in fact, the subject is complex enough that it has not at all been boring. So I'm going to talk to you a little bit about rice blast and mostly about the emerging disease, wheat blast. So these blast diseases are caused by a fungus known as Magnaporthe oryzae. You will also hear it called Pyricularia oryzae. So rice blast is an ancient disease. As far as written history goes back, you can find references to this disease. Whereas wheat blast is an emerging disease, as you know, first reported in 1985 in Brazil. So, uh-oh, I can't change now. Okay. So these are important diseases, but there are many plant diseases that threaten our food. So here you see rice blast. This is a particularly bad field where every panicle in this field is dead, like we often see with wheat blast. It turns out that the super hybrid rices in China are many of them very susceptible to rice blast. The other very famous disease, of course, is the Irish potato late blight, which is responsible for the potato famine in Ireland. So these are both ancient diseases. We've known about them a long time, but unfortunately, we're still dealing with them. However, fungi and mycetes, the eukaryotic filamentous pathogens, are highly adaptable, highly variable. So new diseases keep cropping up. And here's the case of wheat stem rust caused by a fungus, Puccinia graminis tritici. And it had pretty much been controlled for quite a while. But then a new race, UG99, appeared, which had overcome all of the resistance genes that we were using to control the disease. And so this was a scary situation and caused a lot of research for trying to get this under control. Banana fusarium wilt, I love bananas. This disease is a new race, TR4, which threatens our banana production. And so it's also something that needs a lot of research and attention. Okay, our topic, of course, is wheat blast, caused by the fungus Magnaporthe oryzae triticum pathotype. So you're going to learn that Magnaporthe oryzae comes in several lineages or families that are specialized on particular host plants. So in particular, wheat blast is hard to control because theoretically, at least, and I think it's true, one spore, one infection can kill the entire wheat head. And so that's pretty hard if you have to prevent every single spore from infecting a plant. That's pretty hard to do. We only have one useful resistance gene, which we'll talk about, and that gives partial control to the most serious form of the disease, which is head blast, which you see here. These are pictures, these are all pictures from Bolivia. In favorable environment, the fungus, the disease overwhelms resistance plus fungicides. So integrated pest management is really important because we don't have as strong a tools as we'd like to control it. And therefore we need to use everything we've got. Okay, the fungus is seed-borne. So it invades seeds And that has huge potential impact on global grain trade because it can ride along with grain as it moves around the world. And in fact, healthy looking seed can contain the fungus. That's the seedborne or grain trade is probably the reason the fungus moved to South Asia, to Bangladesh in 2016, and then to Africa in 2017. So the wheat blast fungus is on the move. And here you see a field in Bolivia where warm, rainy weather at heading results in 100% empty heads. So here's an outline of the talk. We're going to first talk about blast disease biology because it's important to know your enemy. And by understanding how the fungus causes disease, maybe we can find that Achilles heel to stop the fungus from succeeding. Then we're going to talk about the genomics of the blast fungus. So this is important for understanding pathogen variability and evolutionary potential. And then we'll talk about a little bit about the genomics of the host and strategies for disease control. Okay, so I started out by contrasting the ancient rice blast with the new emerging wheat blast, and here's some interesting contrasts. So rice blast is caused by different population, a different lineage or pathotype, which we refer to as Magnaporthe oryzae, or MOO. It's mainly asexual reproduction in the field. There's a lot of research on rice blast. It's probably one of the very best studied plant pathogens because it's been a model system. And so we know quite a bit about how infection occurs. Because the disease has been so important, breeders and geneticists have identified over 100 major rice resistance genes. And 38 of those have been cloned and characterized at the molecular level. So we have those in our hands to use as tools. These major resistance genes, of course, work because they recognize particular proteins or molecules in the fungus, which are called Avirulence effectors. And 13 of the rice pathogen Avirulence effector genes have been cloned. And we have 11 pairs, which we'll talk about a little more, but examples where we know both the resistance gene and the Avirulence gene. Contrast this new disease on wheat. It's caused by a different population. There's evidence for both sexual and asexual reproduction, which theoretically already makes this harder to control. Pathogenicity mechanisms, we don't know as much about wheat blast, but what we do know is that they appear to be conserved with rice blast. So far, there's only one resistance, and this is the 2NS resistance. It's a partial resistance, very dependent on background, so it's not always effective. We do know that many of the AVR effector genes that have been cloned and characterized in the rice pathogen, many of those also appear to occur in the wheat pathogen. Okay, so here's a leaf lesion for rice blast, and you see this nice speckled appearance, which is due to the spores, the conidia. And here you see the asexual conidia forming, which land on the surface and form this specialized cell, the Appressorium, for punching through the host surface. It's a very intricate process. And what you see here is that the spore is totally empty, because what the fungus does is that it redeploys everything that's in this spore to put it in the Appressorium and to build up turgor pressure. So this fungus is highly specialized for getting into host plants. It's a haploid filamentous ascomycete. It's got a very complex race structure, so there are many races that differ in their ability to infect different rice cultivars And that difference depends on the particular resistance genes in those cultivars. So very, very complex. As I mentioned, there are over 100 R genes that have been identified and 500 quantitative trait loci in rice. But the disease is still very hard to control because of the variability of the fungus. Okay, so we say that Magnaporthe oryzae is a sophisticated serial killer. And this is the spore electron micrograph from my colleague back in the days when I was at DuPont. And you see this little glob of substance on the tip of the spore. That's a special mucilage, which is in the tip of the dry spore and explodes in wet conditions to form this mucilage to stick it to the surface. And this is important for the fungus because infection requires high humidity and freestanding water. So the fungus doesn't want to wash off of the leaf. Again, you see this Appressoria where all of the contents of the spore have been degraded by apoptosis and moved into this cell, which is now glued very firmly to the surface and which produces a penetration peg to punch through the surface. This fungus builds up extremely high turgor pressure, 80 times atmospheric pressure, which is about like the pressure in one of these racing bicycle tires. So it's very high pressure for a biological organism. And it also produces powerful adhesives to glue itself to the surface while it exerts that pressure. So a key factor, and one of the things that gets a lot of study in rice blast, is effectors. Effectors are pathogen proteins that are specifically expressed and secreted inside host tissue. So when the fungus is growing outside the host, it doesn't express these genes at all. So here you see the spore that's already emptied out with an Appressorium that produced the penetration peg. The fungus then grows inside the cell lumen as this specialized bulbous invasive hypha. And as it grows inside this lumen, it is surrounded by host plant membrane. So it actually invaginates and recruits host membrane to surround itself in a protective shield as it grows inside the host cell. So we're going to hear today about the two opposite roles of effectors depending on host genotypes. So mostly the job of effectors for the fungus is to promote host susceptibility. But in cases where the host has a resistance gene that encodes a protein that's a receptor that recognizes that effector, then it's an Avirulence effector and triggers host immunity. So there's these dual roles. So just to show you some of the cell biology that we're doing in my laboratory, we're doing a live cell imaging of the fungus as it invades host cells. And just to show you that effectors accumulate in two patterns around the hyphae inside the host cell. So first you have extracellular effectors, and I mentioned to you that hypha is surrounded by plant membrane. So the effectors that are secreted extracellularly that stay outside the host plant cell surround the hyphae, and you get this nice outlining pattern with this green the effector that's labeled with the green fluorescent protein. However, actually most effectors from the blast fungus are translocated inside living host cells, where they actually move for long distances ahead of the fungus to prepare those host cells. So this is really quite a stealth pathogen once it gets inside the cells. So the cytoplasmic effectors accumulate in this specialized interfacial complex, which we call the biotrophic interfacial complex, and they're translocated inside the host cytoplasm to turn off defenses and to do other things the fungus needs to do. So we don't know, we guess there are hundreds of these effectors that go inside the host cell. Okay, but this is the other function. Avirulence effectors trigger resistance. So as I mentioned, we've cloned 13 AVR genes, and 11 of these correspond to cloned rice resistance genes. There are over 100 major resistance genes in rice, and many more AVR genes remain to be identified. But we can't reliably control the blast fungus because it's able to overcome or break individual resistance genes within 2 to 3 years of their deployment in the field. And this often happens by deletion of the gene from the fungus. So those of you in plant pathology know this famous representation of the gene for gene hypothesis from Harold Floor. And in fact, avirulence effector genes from the pathogen are either, for this pathogen, they're either present, so they're dominant or present, or they're absent or recessive in the case of a diploid fungus. The host has a corresponding resistance gene, so they're pairs. Think of this host resistance gene as encoding the receptor that recognizes this protein from the fungus. And when it recognizes that protein, it says, oh, there's a fungus, turn on defenses. And you see this very, very effective hypersensitive death of the first cell. However, host cultivars that lack that resistance gene can't recognize the AVR effector, of course, and so the fungus succeeds. Or in our case, if the fungus deletes this AVR effector gene, it's now able to infect even the host with this resistance gene. So this is the game we've been playing. Breeders introduce resistance genes into varieties. The fungus mutates often very, very quickly to escape that recognition and to overcome the effect of that resistance. So breakdown of our genes in the fields comes from mutation, mainly deletion of the corresponding AVR gene in the pathogen. This is just another closer up view of the fungus with the avirulence effector, AVR pita. And this, in this case, the rice variety does not have the resistance gene pita, and the fungus is growing happily, whereas the same fungus in a variety with pita, you see the cell death. And so these major rice resistance genes are very strong, very complete resistance, which we call major resistance. So pathogen AVR effector genes are key to blast Magnaporthe oryzae biology. They determine rice cultivar specificity. The presence of the AVR effector gene triggers resistance, whereas the absence of the AVR gene results in susceptibility. So especially these AVR genes are highly mutable. So normal mutation frequencies in a gene is around 10 to the -6, but often AVR genes mutation frequencies are up around 10 to the -3, and they're often just totally deleted. They're totally gone from the fungus. So this is responsible for the rapid ability of the fungus to overcome diploid R genes. And I'm not going to have time to talk today, but these same AVR effectors control host species specificity in the blast fungus. So let's move to talking about genomics of the blast fungus, understanding rapid pathogen variability. In particular, why do we have so much trouble controlling it? Why is the blast fungus so fast to overcome resistance genes that are deployed in the field? And so I just want to stop as a more senior scientist, as a graduate student, I couldn't have imagined genome sequencing of anything. It was just totally beyond imagination. And now with next generation genome sequencing getting better and better and better, it's impacting all of our lives. So I'm showing this Ancestry.com because people are getting their own genomes sequenced in order to understand where they came from, where their ancestors are from, what properties are in their genome that might make them susceptible to diseases or other things. So in this case, we have this weekend, Father's Day. And so Ancestry.com is saying, give your dad a gift of their genome sequence. So this is really an amazing occurrence. So I wanted to just talk about that as something that's in our everyday lives, but genomics has changed agriculture as well. And I want to just talk here about different sizes of genomes. So we're working with a fungus, and typical Ascomycete fungus has around 40 million base pairs in its genome, and it's around 12,000 genes. So that's relatively small, although it's much, much larger than bacteria. So plants are really variable in their genome sizes. The rice genome, for example, is 430 million base pairs. It has around 40,000 genes. The human genome is around 3,000 million or 3 billion base pairs with over 20,000 genes. And then we're talking about wheat today. Wheat is probably, it's among the biggest of them all because it has 17,000 million base pairs, so 17 billion base pairs in its genome with 100,000 genes. Wheat, of course, is a very complex crop because it's got 3 different genomes together. So you have 6 copies of each of the genes that you might be interested in. So there's a lot of differentiation in the sizes, but genome sequencing is now getting so big that it's easy even to sequence and assemble wheat genomes. And so many, many varieties of wheat are also now being sequenced. Okay, so here's a family tree for Magnaporthe oryzae, which is one species with crop-adapted pathotypes or lineages. So I want to point out that this tree is based on whole genome sequencing of many, many strains, more than are shown here. And the markers are single nucleotide polymorphisms. So there's single nucleotide changes. In particular, we call these SNPs, and it's based on SNPs per megabase of uniquely aligned DNA. So as you know, all organisms have a lot of repetitive sequences, lots of transposons, but we only look at the nucleotide changes in the single copy DNA. So here you see the ancient members of this family. So rice, caused by the oryzae pathotype. Foxtail millet blast is very serious. That's still a staple grown in some parts of Africa and Asia. It's infected by the Soteria pathotype. Finger millet, grown in Asia and also Africa, is affected by the Eleusine pathotype. So the family, although this family infects most grasses, there's host specialization. So all of these crops, all the diseases on all of these crops are predicted to have arisen around 7,000 years ago. And in fact, we think that rice blast came from the Soteria disease. However, each of these blast diseases is still the most important disease for each of these cereal crops. So we haven't managed to control any of these diseases. On the other hand, in the 1980s, we had two new diseases. So you know about wheat blast here, which was first reported in Brazil in 1985. But a new disease happened on ryegrass, in particular, used for turf, for lawns, and also for forage crop. And this occurred, a new disease occurred in the United States in 1992. So it's interesting that both of these diseases came about the same time. So you see the tree and you see the genetic distances here represented by, or the numbers of single nucleotide polymorphisms represented by the distances between strains. Okay, so I wanted to just mention, but we don't have time to talk in detail, but we've learned that Magnaporthe is divided into pathotypes specialized for different grasses. These pathotypes differ by avirulence genes. So avirulence effector genes, just like we've talked about for rice pathogens, that block infection of other grass species. So theoretically, and we have evidence that loss of pathogen, or in particular, loss of an avirulence gene from lolium strains is what led to the creation of the triticum lineage in Brazil in 1985. Emerging wheat and ryegrass populations show higher levels of genetic divergence than ancient populations infecting rice and millets. So maybe it's just because they've had a shorter time to adapt to their host. But it's really quite a striking effect, because here you see this little bubble represents many, many rice pathogens from all over the world and from different decades. Whereas here is the wheat lineage and here is the ryegrass lineage. And if you look at nucleotide diversity here, you see that you have higher nucleotide diversity in the ryegrass lineage, but especially in the wheat lineage. So there's really extraordinary variability in the population we're dealing with in wheat fields. So why does increased variability in the wheat blast lineage matter? It matters because different strains show different levels of aggressiveness towards wheat. So some strains, in particular, the early strains from the 1980s and the early 1990s cause serious disease, yes, but they're not as serious. The disease is not as serious as the disease caused today. So different strains show different levels of aggressiveness towards wheat, which means some strains, or the more recent strains, cause worse disease faster, which is bad news for us trying to control this disease. So why is this true? Different strains infected different secondary host plants. So I've told you that the fungus is specialized to a particular crop, but there are alternative hosts. And this is important because the other plants serve as reservoirs for the pathogen. And so it'll be, for example, in Bangladesh, the farmers thought they could get rid of wheat blast by burning the wheat fields, and they did that. but the pathogen did not help to control the pathogen, and it likely was hanging out in other host plants beside the field. So we need to understand this, and also need to understand what other crops might be at risk. Different strains have different levels of fungicide resistance. We need to know this when we're using fungicides. And some strains in the ryegrass lineage also cause disease on wheat. So I mentioned to you that we think the wheat lineage did evolve from the ryegrass lineage. And in fact, some of the ryegrass pathogens that are growing in the United States right now can infect wheat. They're just not as aggressive as the wheat pathogens. So what accounts for this variability? Okay, what you need today is a reference genome to use as a tool for then sequencing many hundreds of other strains or varieties of whatever organism you're working with. So working together with my colleague at Kansas State University, Dr. Sanjian Liu, we've produced a reference genome, which is telomere-to-telomere genome assembly for an aggressive Bolivian wheat blast strain that we've worked with a lot, which is named B71. So just like rice pathogens, B71 has 7 core chromosomes, which you see labeled around the edge here. So you've got these chromosomes laid out in this circular pattern where you can look at repeats and various features of the genome and various types of genes and just compare. So all strains, rice pathogens, millet pathogens, wheat pathogens, and ryegrass pathogens have 7 core chromosomes. But this sequence actually gave us the first view of dispensable mini chromosomes. So these are supernumerary chromosomes. They're in some strains and they're not in others. Some strains have up to 4 copies, maybe more, and some just have 1 or 2. So these are smaller, which is why we call them mini chromosomes. They're highly variable in sequence. And interestingly, they seem to be in dynamic equilibrium with the ends of the core chromosomes. So for example, 2 of the genes I've studied most are in rice pathogen core chromosomes, different core chromosomes. In the wheat pathogens, they're on a mini chromosome and they're sitting right next to each other. So this is a vehicle for variability and for moving genes to new places and for retaining genes that are deleted from the core chromosome. So I think this is really a significant bit of biology. I want to just point out it's been known for a while, and this is with AVR PETA from rice pathogens, which corresponds to the PETA gene in rice, which has been periodically deployed all over the world in rice crops. So the amazing finding from Dr. Yukio Tosa and colleagues, Izumi Chuma, is that the AVR PETA gene doesn't exist in just one place in the genome. It's all over the place. So this gene, the AVR PETA gene, is on chromosomes 1, 3, 4, 5, 6, 7, or on dispensable mini chromosomes in different strains. So as you know, if you look at housekeeping genes on chromosomes, you expect them to be in the same place, with few exceptions where there are major rearrangements. But AVR PETA, and now we know a bunch of other AVR effector genes, are moving around in the genome. And so this just shows you can't see all the detail here, but the gene has been found in 4 different places in chromosome 4. The gene is this little red box. And so all you have to see that the patterns surrounding that little red box are very different in the different strains. They're in different places. And the star here represents the chromosome ends, the telomere sequences. So in some cases, that gene is right next to the telomere. So this is really amazing behavior for genes. So AVR genes corresponding to periodically deployed R genes in rice are changing locations in the fungal genome. And so this is a work in progress, but we think that this is what's happening as the fungus is overcoming resistance. The fungus has an AVR gene, and it's avirulent on a rice cultivar with the corresponding R gene. So the fungus just deletes this AVR gene. It's totally gone. The fungus without the AVR gene is now a pathogen of the rice cultivar with the R gene. However, when you remove that cultivar from the field, the fungus appears to regain that AVR gene, but in a different place. And so this is the working hypothesis that many chromosomes serve as effector gene reservoirs, so they keep genes around that have been deleted from core chromosomes, and they serve as mediators of horizontal transfer through the fungal parasexual cycle, which we don't have time to discuss today. But in other words, the fungus can throw away a gene and then get it back when it doesn't need, when it's not faced with a rice variety with the corresponding R gene. So that's pretty remarkable behavior that we need to understand. And that helps to explain why this fungus is so quick to overcome resistance genes we deploy. So I want to just mention that wheat blast pathogens have copies of many of the AVR genes that correspond to known resistance genes in rice. And here you see our same circle with B71 chromosomes, and here are the telomeres. And these are genes in the wheat pathogen that correspond to known effector genes, including AVR effector genes in rice pathogens. And so this led us to the hypothesis that cloned rice resistance genes will work in wheat to control wheat blast strains with the corresponding AVR genes. And so Monica Navia Arrutia worked on this, and she just obtained her PhD last year, asking the question, will rice resistance genes function in wheat to control blast disease? She screened 102 wheat pathogens for the presence, absence of AVR effector genes corresponding to the genes we know about in rice. She demonstrated that one of these, AVR PIZT, in the wheat pathogen T25, is functional. So that means if you put it into, transform it into a rice pathogen, and now ask that rice pathogen to infect rice containing the corresponding gene PIZT, it mediates recognition and resistance. So it should work with the rice resistance gene. So Monica produced transgenic wheat plants expressing PIZT. This was really hard work and really well done. But the bottom line is we were disappointed that it failed to achieve useful levels of resistance to the strain T25. So we need to think about whether this strategy is good for pursuing in the future, since not all strains have the functional AVR PIZT gene. At best, this is going to have partial resistance on only a subpopulation of the wheat blast population. So this was a proof of concept experiment that didn't work. There could be other rice resistance genes that work better. So I want to move on now to genomics of the host and strategies for wheat blast control. So I'm going to briefly review the current status. Can we find new effective resistance genes? And I'm going to end by wondering if gene editing strategies will work for wheat blast. We know they work for other diseases, and I'll show you that, but we don't know yet about wheat blast. So again, we're back to our comparison. And while we focused mostly on rice blast, we know that wheat blast is caused by a different, highly diverse population. We know that wheat blast, there's evidence for both sexual and asexual reproduction. We know that the mechanisms of pathogenicity appear conserved. So if we could find out something along those lines that would help rice blast, that could help with wheat blast. But in contrast to rice blast, where there are over 100 major rice resistance genes, we only have one resistance, the 2NS resistance. And as I mentioned, this is a partial resistance that's really dependent on the background of the wheat variety it's in. But as we've learned already, many cloned rice AVR genes are conserved in the wheat pathogen genome. Okay, so in the United States, we don't have the triticum lineage, and we're afraid of it. So all of the experiments that we do are in biosafety level 3 laboratories in the US. Here you see Christian Cruz. Now, Dr. Cruz, who's an assistant professor at Purdue University, and he's all protected from spores of the fungus, and he's spraying spores onto wheat heads in our BS level 3 laboratory. This laboratory is very contained, and you can't leave that laboratory without taking a long shower and washing your hair and just making sure you're not transporting fungus out of the laboratory. So we're focused on head inoculation, screening winter and spring wheats and wild wheat relatives, and we score infection by percent of blighted wheat spikelets. And that's what this looks like. So here's disease severity, percent infected spikelets. And you see 5% here, there's one lesion here and another one here, I think. 70%, so most of the individual spikelets have disease, and 100%. The neck of the developing head is very susceptible, and if infection occurs here, then everything above that point is dead from blocking of photosynthesis. Okay, we all know that laboratory studies are important, but they're not definitive. And that's where we've had a really extremely valuable collaboration here in Bolivia. So we've been working between BSL-3 laboratories. I should mention there's a second laboratory in the US that we're also collaborating with at the USDA Fort Detrick facility. And we're doing field tests with collaborators here from ANAPO and CIOT. And here is our group. Here is Christian Cruz, and here is Gabriella and Lydia. And I'll mention I'll mention more later. So we do 2 test cycles per year, winter cycle in Santa Cruz, wheat production areas, and summer cycle in Quiricias blast hotspot, although actually we've moved on from Quiricias now. I didn't change the slide. Okay, so phenotyping winter and spring wheat varieties. In Kansas, we grow winter wheat, so that's what we care about. And in fact, here you see the disease severity scale that I've explained to you already. And these are just different wheat varieties. And you see there are some that are really pretty good. They're 0 here or below 5%. And then you have increasing levels of susceptibility. Spring wheats are much more susceptible. So there's just a few that are really low, and the curve towards susceptibility goes up very quickly. Okay, so this was a surprise. We didn't expect this continuum of susceptibility based on rice blast. But I can tell you now, although we were hopeful at first, much of this resistance is due to the 2NS translocation segment from Agilops ventricosa. So we thought we had a lot of sources of good resistance, but in fact, it turned out to be mostly just one source. So that work that was proven here in Bolivia in tests by our team. The identification of a wheat blast resistance gene on the 2NS translocation fragment of Agilops ventricosa. So this 2NS segment, which has been brought in from wild wheat, was brought in because it has rust resistance. So it actually has 3 rust resistance genes and 2 nematode resistance genes. So lucky for us, it was pretty widely deployed already within the wheat germplasm. We already had molecular markers. And so field tests on 2NS near isogenic spring wheat lines really proved that the 2NS does confer resistance. Reductions occurred in all 10 comparisons over 2 years and 3 experiments with an average of 85% reduction. And so here you see the line without 2NS and the isogenic line with 2NS, and you get a pretty major effect. So that's good. It's also good that this resistance seems to be holding up with the more aggressive isolates. So for example, we have 2 strains here. T25 was from 1988 in Brazil, B71 from 2012 in Bolivia. And so you see cultivars without 2NS. You see that the blue bars here are strain T25, whereas the red bars are strain B71. So without 2NS, everything seems to be pretty susceptible. Cultivars with 2NS, their B71 is more aggressive. The red bars here are higher than the blue bars, but it still has an effect. So that's the good news. So a lot of work has been done, and now in increasing numbers of labs, screening for resistance in various other cultivars and also in wild wheats. And I would have to say now a lot of our hope is in wild wheats, although we still think that some of the more resistant varieties with 2NS might have other genes that are having additive protective effects. But this is just some data from Giovanna Krupe, who got her PhD with me and with Christian Cruz at Kansas State a couple of years ago. And this is her experiment in the BRI of various Agilops tauschii. And again, you see a lot of them are totally susceptible, but there are a few, and we're hoping, since the 2NS came from a wild wheat, that we'll find different loci from wild wheat. So I want to talk to you briefly about, so I see it's going faster. I hope the time's okay. Want to talk briefly about genome-wide association study, because genome sequencing, as I said, genome sequencing of wheat's doing great now, although it was the last crop sequence because it's so large. So GWAS is a strategy to identify associations between markers and traits linked based on linkage disequilibrium between that marker and the phenotypic polymorphism, whatever it is you're studying. So this is an alternative to doing biparental populations. You can use natural populations. It eliminates the need for developing biparental populations, accounts for historical recombination, higher mapping resolution with smaller genomic region intervals. So it has, it's not perfect because it it isn't able to detect small effect genes. But here you see the genotyping, in our case, head blast resistance, phenotyping of head blast resistance. Genotyping, again, we're using single nucleotide polymorphisms. And with the help of computers and lots of work on people's parts, people are able to correlate the phenotype with various SNPs. So here's Giovanna's experiment, where you have 384 wheat lines were phenotyped with strain T25, and genotyping by sequencing was used for identification of the single nucleotide polymorphisms. And this just shows a physical map of the positions of the 13,175 SNPs that Giovanna used, with this heat map showing the density of SNPs. And here's the major output, which is a Manhattan plot of wheat spike blast severity. And you see this dotted red line. Everything above that line is probably significant. And basically, Giovanna and others are doing this mostly find the 2NS resistance. She found a couple of other things which probably aren't that useful. But 2NS is really the main thing we're coming up with. So the last thing is CRISPR-Cas9 engineering for plant genome editing. So everybody's excited about plant genome editing. I should say that there's other systems besides CRISPR-Cas9, so there's lots of potential. But very, very briefly, you have the genomic sequence that you want to change, and you have a target-specific guide RNA sequence in a special construct. And these days, people just transform the Cas9 protease, it's a protein, into the variety with the guide RNAs. And the Cas9 protein cuts the genome at that particular locus. And then you get mutations based on how you repair that double strand break. So specifically, genome editing relies on break and repair of DNA. So you have a nuclease-induced double strand break, and we can target it to the locus of interest. And the cell decides how to fix it. So the cell can do non-homologous end joining, where they just put the pieces back together again, but there's always something wrong, various changes where they're put back together. So variable length insertions or deletions. On the other hand, sometimes the cell could do homology-directed repair. In this case, you give it homology, and the cell uses that homology to repair the break and to put in the sequence you want to put in. So practically speaking, non-homologous end joining is now very efficient. It's efficient in wheat, and it's used for eliminating gene function, because as we said here, it just causes changes and will change. If it's in the gene, it's going to disrupt that gene. So what you need are susceptibility genes. These are the opposite from resistance genes. You need a gene where if you destroy the function of that gene, it stops infection. So susceptibility genes are a brand new topic that a lot of people are focused on. These are plant host genes that are essential for disease. And once you break those genes, you can stop the pathogen. So this strategy is efficient now. The homology-directed repair is currently not efficient. It needs work just because it's not working as well to put the precise sequences in that we'd like to be able to put in. So for proof of concept, I've jumped to a different system in wheat. So simultaneous editing of homeoalleles of the wheat MLO genes in hexaploid bred wheat confers resistance to powdery mildew. So it's been known for decades that MLO is a susceptibility gene. So if the wheat has the wild type gene, it's very susceptible to powdery mildew. But if it has a mutant gene that's not functional, it now becomes resistant. So this is a classical susceptibility gene. And so in this study in Nature Biotechnology, they very patiently used genome editing to change all 6 copies of the gene. And here you see an assay of powdery mildew fungus microcolonies per total number of germinated spores. So how efficiently are the germinated spores infecting wheat? And you see that you have pretty good infection if you just take out the AA or the BB or the AABB, but you need to take out all 6 copies, and now it becomes resistant. So the wheat susceptibility gene, MLO, is critical for powdery mildew infection, and it works after knocking out all 6 copies in wheat. And this just shows some pictures of that. Here's the fungus. The fungus grows outside the host plant, so you see these hyphae, and you see in the AABBDD genome edited variety that the fungus just isn't growing very well. And you see that it looks pretty healthy. So that says it could work. Unfortunately, MLO resistance is specific for powdery mildew disease, and susceptibility genes identified for wheat blasts are only associated with minor effects. And so far, this hasn't resulted in anything for control of wheat blasts, but we need to keep trying. So I'm going to end by saying what is needed to control wheat blast disease. We really need more resistance genes. We need effective resistance genes to stack with the 2NS genes. And in particular, I think we're going to need a lot of effort to explore wild wheat relatives. We need research to identify wheat blast susceptibility genes with major effects so that we can knock those genes out by genome editing and impact wheat blast disease. We need understanding of the mechanisms of variability and the evolutionary potential of the wheat blast fungus, because this fungus is still evolving and we need to have some idea where it's headed to. And we need a lot more research on basic disease mechanisms. So I've talked to you about wheat blast, and of course, you know, this is only one of many, many threatening diseases we need to get control over. So the world really needs to decide to do research on emerging plant diseases, and not just emerging diseases, but old plant diseases that we can't control very well. So we need a whole lot more resources into research. So I'm going to just briefly point out Dr. Liu, who is involved in the bioinformatics and genome sequencing. Dr. Harold Trick, who's a wheat transformation expert and helped Monica. Dr. Stack, whose work I haven't been able to talk about today, but he's done really nice work on plant diagnostics with Javier Quiuna, who's here now in Bolivia working on wheat blast, and Christian Cruz is now at Purdue, and Gary Peterson and Carrie Pedley and Mark Farman. So I want to just say special thanks to our collaborators in Bolivia. It's been a really productive collaboration, but also really just fun and a good experience. So I want to say special thanks to Gabriella, who's my host here today. And here she is in the field. I want to say special thanks to Lydia Calderon, who's an expert on phenotyping wheat blast and scoring wheat blast symptoms and every other aspect of studying this disease in the field. And Javier got his master's degree, I think a couple of years ago at Kansas State University. And here he is with his advisor, Dr. Jim Stack, much more formal than you typically see him. And so he's here now in Bolivia leading our wheat blast collaborative effort, leading it leading the ANAPO, CIAT, KSU, USDA wheat blast collaborative effort. So thanks to everyone. And with that, I'll take questions.
Thank you so much, Barbara. Okay, it was a great presentation for me because you explained plant pathology for one hand, and the other hand, you explained the genomics. And for us, it's very important to know these tools. Okay, we have different questions now. First, well, we had a question from Maria Mercedes Roca. She says, great talk, Dr. Valent. And she had a question. Is it possible to dream that one day we could design specialized and more specific synthetic genes like the AVR genes that may confer resistance to pathogens? Or that genomic editing might be used to tick host resistances?
Yes, I think it's very possible. And I think we shouldn't give up. We should keep working on that. So there are people who are trying to improve rice resistance genes or resistance genes in other crops. Specifically, you have a resistance gene with a certain specificity, but there's other strains of the pathogen that have other forms of the AVR effector. So can you evolve that R gene so that it recognizes those other forms as well? And so there are preliminary experiments trying to improve the R genes we have. And I think that genome editing is really an exciting potential for our future. For one thing, it's not considered GMO technology in many parts of the world, so it's going to have easier approval. And we just need to study it and figure out what genes to target. So the technologies are there, and what we need is knowing how to get to where we want to be. But yes, I think that's the future. I think we need research on that.
Okay, thank you, Barbara. And we have other questions. It's from Betsania. She asks, how many and what are the genes involved in resistance in wheat blast?
So in wheat blast, we really haven't, so we know that there are some genes of the type that we find in rice blast. So Dr. Yukio Tosa in Japan has isolated AVR genes and corresponding resistance genes. So right now it's maybe up to about 9 resistance genes that have been identified. The problem right now is that the blast population in the field is overcoming those genes already. So that's an important question, and it's a real puzzle to me that there are so many R genes that we find in rice, and we're having so much trouble finding them in wheat. So it could be they're there, but the fungus doesn't care as much. It's a really important question, but we need more resistance genes. So we need a lot of effort to find them.
Okay, I had other questions. Will the damage caused by wheat blast be greater than the causes by Pyricularia? Okay, I think the question is about wheat blast and rice blast. Could you see a difference of the damage on the field or on the crops?
So wheat blast, actually, So we have more resistance in rice. So I would say wheat blast, there's more damage, but I showed you that field of panicle blast in rice. That's just as bad. Every panicle in that field was dead. So that was a highly susceptible variety. Another puzzle is that with rice blast, the crop is vulnerable at every growth stage. So leaf blast is can be very deadly. But that's not been true with wheat blast. We hope it doesn't change. So far, the wheat plants, the leaves have not been as susceptible to the fungus. And that makes the period of danger smaller. So we really hope that the fungus doesn't adapt to cause more disease on leaves. But in rice, it's a bad disease on leaves and seedlings. And so that's also a problem with rice.
Yeah. Okay. I have other questions because, for example, first we found wheat blast here in South America. Then in 2016 in Bangladesh, then Pakistan, and the last year in Zambia. What do you think? Do you think that we are near to identify strategies to develop some tools to fight with blast? Do you think?
So we're still depending on integrated pest management. So that includes adjusting planting date, because if you can have heading occur in dry weather, you're okay. And we do have some resistance, but we need resistance to stop it. So in Bangladesh, the first report was extensive, many fields over many different districts. And it spread the next year, even though the weather wasn't conducive. So right now, I don't see anything stopping it. We don't know about Zambia, but I guess I'm worried. What we need to do, so this is something that Javier worked on. We know that this fungus is moving through grain, through seed, either for planting or grain trade. And so we need to have diagnostics that can detect it and stop it. So I think our main strategy should be stopping it from establishing, because once it establishes, it's there, and we don't know whether other plants it's on that it's hiding on. So it's very hard. So Javier worked on diagnostics for seed, and we still have challenges there in terms of the quantities, the sensitivity of the detection. But we need to be careful and keep it from spreading beyond where it is. And that's going to be hard.
Okay, Barbara. Okay, thank you so much. For me, we feel very grateful with you because it was a great presentation, and thank you for the support. Okay, Cecilia, please. Thank you, Barbara. Thank you a lot.
Thank you.